Good morning, everyone. Today I'm going to discuss the antibacterial agents, their mode of action and resistance. So, drugs have been actually used initially for the treatment of certain infectious diseases since the 17th century, starting with the quinine for malaria, which is a parasitic disease. However, chemotherapy as a science began in the last decade, in the 20th century, and it was with the understanding of all the principles of selective toxicity, because antibiotics, when they are going to be delivered, they need to be inhibitory and killing to bacterial cells and not to the host cells. And second, the specific chemical relationships between microbial pathogens and the drugs should be well taken into consideration. And also, most importantly, the development of drug resistance that has emerged after overuse and abuse of administration of antibiotics, and the role of combination therapy that was administered in situations where the bacteria has acquired resistance to the antibiotics. Now, Alexander Fleming is the one that discovered initially penicillin, and that was by accident. And this penicillin is released from a mold called penicillium. Now, the current era of antimicrobial chemotherapy began in 1935, and discovery of antibiotics like sulfonamides was in 1932. So what did Fleming do actually? He was working on Staphylococcus aureus, and Staph aureus was grown on blood agar plate. So Fleming left the plate on the bench for a couple of days, and then when he came to look at it and work on the organisms, he found, as you see, Penicillium chrysogenus, which is down there. And this is a fungus that actually was grown. Now, at the place where the fungus is grown, as you see, there are no colonies of the bacteria of Staphylococcus aureus. So this means that something has been diffused from the mold in the blood agar plate that is inhibitory to bacterial cells. And from here came the idea of drugs that inhibit the growth or kill bacterial cells, and these were called antibiotics. So we can see that the sources of some common antibiotics and semi-synthetic ones, are coming initially either from fungi or from bacteria. Now, the general mechanisms of action of antibacterial agents lies in different areas in the bacterial cells. And we can see that we have certain antimicrobial agents, or let's say antibacterial agents that we also call antibiotics, that act on the cell wall synthesis, like for example the beta-lactams, cephalosporins, and carbapenems. We have those like metronidazole that act on DNA replication of the cell, and in this way they can stop DNA replication and kill the cell. There are fluoroquinolones that act on the topoisomerase enzyme that is important in bacterial replication. Sulfonamides, trimethoprim, acts on nucleotide biosynthesis within the cell. Now we have other other antibiotics like the peptide antibiotics, which act directly on the cytoplasmic membrane integrity, and they will disrupt the integrity of the membrane. Others like rifampin, and this is mainly given to Mycobacterium tuberculosis as one of the antibiotics treating tuberculosis. It acts on RNA transcription. And finally, we have the glycyl cyclins like aminoglycosides, macrolides, and others. These act on protein synthesis. So you see that we have different types of antibacterial agents that act on different functions of the bacterial cells, and when they act on these functions, then they will stop bacterial replication. This is actually disrupted by the action of beta-lactamases, which are produced by bacteria and causing resistance to penicillins. We will talk about this later. Inhibition of protein synthesis, as you can see, we have a number of antibiotics that act on different areas in the ribosomes. So we have the chloramphenicol, for example, that binds to the 50S ribosomal RNA, and it inhibits the formation of peptide bonds. While down there we see erythromycin binds to the 50 Svedberg unit of the ribosomal RNA and prevents the movement along the messenger RNA. Tetracycline, as seen, interferes with the transfer RNA anticodon reading of the messenger RNA, and in this way the RNA won't be able to proceed with its function so that protein synthesis can occur. Now, streptomycin changes the shape of the 13S, as you see, ribosomal RNA, and in this way causes the messenger RNA to be read incorrectly. Now, inhibition of the cell membrane functions, as we said, like the action of polymyxins. These ones, they act on the outer membranes, and they disrupt the integrity of the outer membrane, leading to cell damage and death. And this is caused, the antibacterial agent that acts on cell membrane are the polymyxins and colistins. And these antibiotics actually are all drugs. Okay, they have some kind of toxic effect, especially on the kidney. However, due to resistance to all antimicrobial agents available on gram-negative bacteria, recently colistin was the last resort, given singly or in combination with other antibiotics, in order to be able to treat the patients. Now, inhibition of nucleic acids is done by quinolones, and quinolones actually inhibit nucleic acid replication and transcription. Now, Whenever we talk about an antimicrobial agent in general, and here we are talking about the antibacterial agent, it is very important that we take certain things into consideration. That these antibiotics should be readily available, found in all pharmacies at all times, and at a reasonable price that can be affordable to all. Most importantly, this antibiotic should be chemically stable. And of course, before allowing this antibiotic to be used clinically, it is assessed and approved by the Food and Drug Administration in the USA, and there are other agencies and other places in the world that also do the same job. So stability, chemical stability, non-toxicity to the host cell are two major important things to take into consideration. The antibiotic also, one feature is to be non-allergenic. So whenever you go to the doctor and you are subjected to a given procedure or surgical intervention, the nurse asks you about whether you are allergic to a particular drug or antibiotic. So if you are allergic to it, then it has to be replaced by an alternative antibiotic. So selective toxicity is very important. Selectively toxic against a wide range of pathogens to be taken into consideration. Now, the spectrum of action of antibiotics can fall into two categories. Those that are broad spectrum antibacterial agents, and these are active against both gram-positive and gram-negative organisms. Now, the narrow spectrum antibacterials have limited activity, and primarily they are only used against particular species of bacteria. So over here we can see examples of the spectrum of action. Now, the routes of administration. Antibiotics can be given topically in the form of creams, for example, and these you apply them on the skin. Oral administration in the form of pills. Intramuscular administration, so you have injections that are given intramuscularly, and there are antibiotics that given intravenously. So intravenous administration is mostly for severe types of systemic illnesses. Over here we can see that the relative concentration of drug in blood is the highest in intravenous type of administration of the route of administration of the antibiotic, followed by intramuscular and then oral. So safety and side effects, as I mentioned, toxicity is a feature that you need to take into consideration. Drugs may be toxic to different parts of different organs in our body, like the kidneys, liver, and nerves, and as I said, colistin is an example. Allergies, you have certain antibiotics that cause anaphylactic shocks, so this may lead to death. Now, disruption of normal microbiota, and this is a very important feature, because when you are taking broad spectrum antibiotics, whenever there is no need for it, and this way you are taking these antibiotics and you are actually disrupting your normal microbial flora, which is lining your internal mucosa or your external skin. So, in this situation, when you erase or when you inhibit the growth of your microbial flora or kill it by the different types of antibacterial agents that you are taking, you are wiping off the first line of defense mechanisms against pathogens that can invade your system. And this may result actually in secondary types of bacterial infections. And of greatest concern in this situation is hospitalized patients who are debilitated or immunocompromised, and they are found in the intensive care unit, in the ICU. Now, these patients are very prone and susceptible to the effect of external pathogenic organisms which are found in the environment or in devices that you are using. For example, the patient is subjected to a respirator, and the tubings of the respirator may be contaminated by a bacterium that is resistant to all kinds of disinfectants and antibiotics. No matter what you do, it can still be in the tubings. That's why those tubings should be constantly changed. And these organisms will enter into the respiratory system of the patient that is debilitated, immunocompromised, and in this situation they can produce an infection. Though they have a low virulence potential, they can still overcome the immune system found in the host that is debilitated and or immunocompromised. So this also can be entered into the host by intravenous catheters, Foley catheters, and so on at different sites of infection. Now, whenever we have resistance in certain situations due to antibiotics, then the last resort will be to use combination therapy. Try to do combination therapy. And combination therapy options is not an easy thing to do. You cannot just do it by growing the bacterium and putting two antibiotics together on a plate and look at it. No. This can be done by special tests, in order to assess the efficacy of combination therapy. And these tests are called, for example, the checkerboard assay, whereby you can put different antibiotics together and look at the outcome, whether these two antibiotics together are synergistic. Number two, synergistic means when the effect is greater than the sum of the two antibiotics, and in this way you can overcome the infection. Now, if these antibiotics are not synergistic, then they have only additive effect, means indifferent effect. So we can say there is indifference whether you use each one singly or you use them in combination. They are indifferent. They give an indifferent effect. And three, antagonism, when the effect is less than the sum of their independent effect. So in this way, when you give the two antibiotics, you will get antagonism, and in this situation the two antibiotics antibiotics together don't work. Now, how can we assess whether an organism is susceptible to antibiotic or not? We use disk diffusion susceptibility test, or we can use the minimum inhibitory concentration test. And this can be done by the e-test. So the disk diffusion, when you look, you have an agar plate, you grow bacteria on it, and then you put different disks of antibiotics that have particular concentrations. You incubate overnight in the incubator between 35 and 37 degrees, and then you look at zones of inhibition. So you can see over here that there is a zone of inhibition around each disk. Now, for each antibiotic, you need to measure the zone of diameter and compare it to a table to see whether the value in millimeter matches with resistance, intermediate resistance, or susceptibility. The e-test is actually a test that is quantitative, whereby you can have a determination quantitatively of the minimal inhibitory concentration. Now, all these tests will be described fully with their interpretation, and you will be working on them in the lab. Now, in our system, in our medical center, we can actually look at the various antimicrobial profiles by having a kind of a table that is circulated by the Department of Pathology and Lab Medicine, whereby they show you the various values to the different antibiotics and different types of bacterial agents. Okay, and this is actually renewed every year. Now, antimicrobial resistance emerges whenever there are different mechanisms that may induce this type of resistance. We have non-genetic type of resistance, whereby bacteria can replicate, okay? The active replication of bacteria are required for most antibacterial drug actions. So the antimicrobial agent can act only whenever bacteria is replicating, because it is at its exponential stage of growth, and it is in a weak position that can be hit by the antimicrobial agent. Now, microorganisms that are inactive, so they are viable, but they are inactive, that is non-multiplying, so may be resistant against the drugs. As an example, we have Mycobacterium tuberculosis, that whenever it enters into the respiratory system, it goes into the lungs and it forms a tubercle. This tubercle is the sum of all immune cells around the bacteria that engulf the bacteria in order to act against the bacterial agent. However, the bacterial agent remains viable in this tubercle, okay, and non-multiplying, inactive. Whenever there is a situation of immunosuppression by, for example, having chemotherapy or being administered by corticosteroids, which are immunosuppressive, or any other debilitated disease, now the patient become immunocompromised, and the tubercle, which consists of the immune cells, start to liquefy. Once this happens, then the bacteria will start replicating. And when it replicates, it causes an active disease. So at this stage, whenever the bacteria is replicating and producing symptoms, then you can hit it with 3 types of antibiotics using a regimen, okay, and a particular schedule. Now, genetic type of resistance, it can be chromosomally encoded, or it can be plasmid encoded. Now, chromosomal resistance develop as a result of spontaneous mutations in a particular locus on the chromosome. And this type of mutation actually will control the susceptibility to a given antibacterial agent. Now, plasmid encoded, they develop plasmid encoded type of resistance. These are plasmids that harbor particular genes, we call them resistance genes, to particular antibiotics. And these resistance genes, therefore, being on a plasmid, they can be transferred horizontally from one bacterium to another. So this is an acquired type of resistance, okay, which is genetically determined. Now, sometimes those actually genes can be found on particular segments on the chromosomes, and these genes actually may be incorporated into the chromosomes, okay, by translocation of a DNA segment coming from another bacterium by recombination, or for example, the plasmid is recombined within the chromosome and conferring to it a fragment that contains resistance genes. So therefore, resistance genes can be also encoded by resistance genes can be encoded either on plasmids or on chromosomes. Now, what do we mean by intrinsic native resistance? Now, intrinsic native resistance is a naturally occurring type of resistance that is determined by, for example, certain genes that are chromosomally encoded that cause efflux pump. Efflux pumps are genes that will activate certain pumps that will repulse any antibiotic that comes into the bacterial cell, or not allow a target molecule to act, for example, on the bacterial cell. Or you can block the transfer of antibiotic into the bacterial cell. So all these are naturally occurring genes that are chromosomally encoded and that will act on antibiotics. Now, acquired resistance is by horizontal gene transfer, as I mentioned, through acquisition of, for example, genes, plasmids that encode for resistance genes by the mechanism of conjugation that was discussed in genetics, or by transduction that is transferred by a bacteriophage, or by transformation that is acquired by fragments of DNA from disintegrated bacterial cells or by spontaneous. Now, the resistance mechanisms in case of reduced influx of the antibiotics. Mutations in genes coding for outer membranes, for example, the outer membrane proteins, will result in modified structures. So there is a kind of impermeability that occurs in the outer membrane proteins, not allowing the antibiotic to enter into the cell. Sometimes the genes encoding for the outer membrane proteins may be lost. So this will lead again to impermeability of the outer membrane proteins. Increased influx, as I said, by influx pumps. And this is due to mutation gene changes in the genes that regulate the multi-drug resistant pumps. And in this situation, it enhances the efflux of antibiotics from the cell to the outside environment. Modification or protection of the target. So again, mutational changes and the target of the antibiotic occurs, which might decrease its affinity to the antibiotic. And in this way, there is decreased susceptibility. So the antibiotic that enters cannot anymore bind to its target because its target has been mutated, leading to a particular proteins that the antibiotic cannot act on. Or modification or inactivation of the antibiotic. How can this happen? This happens whenever bacteria have certain enzymes that are antibiotic modifying enzymes. And these enzymes actually, whenever they are produced by the bacterium, they either hydrolyze the antibiotic, okay, break it down, or they change its chemical structure. So in these situations, the antibiotics cannot function anymore because they are disintegrated. Now looking here at the resistance mechanism and looking at the things that I mentioned, starting with the efflux pump, okay, and you see how mutational changes in the efflux pumps encoding genes that are chromosomally encoded, okay, will lead to efflux of the antibiotic from the cell. Or whenever you have certain enzymes that are produced, like for example beta-lactamases, and these ones are produced because they are encoded by particular genes, okay, that may be subjected to mutations, leading to the formation of these modifying enzymes that will act on the antibiotic by hydrolyzing it or changing its chemical structure. Now, the other mechanism is by the change in the outer membrane proteins due to mutational changes, okay, of the genes encoding for the outer membrane proteins, leading to conformational change in the outer membranes, leading to impermeability to antibiotics. And as I said, sometimes the genes encoding for outer membrane proteins in the bacterial cell are lost, again leading to impermeability and lack of entrance of the antibiotic. Horizontal gene transfer, as we said, by conjugation, wherever you have the plasmid entering, harboring a particular resistance genes. Transduction, whenever the resistance genes are found on the DNA of the bacterial phage, and this will be introduced into the bacterial cell, integrated into its own genome, and leading to resistance. Or transformation, whenever competent bacteria uptake naked DNA from donor bacterial cells that have been disintegrated. So over here again, we can see how these mechanisms occur by conjugation, transduction, and transformation. Now, as an example of the modifying enzymes that are produced by bacterial cell in order to hydrolyze the antibiotic, we have here the example of the beta-lactamase which hydrolyzes the green beta-lactam ring that you see in the chemical structure of the penicillin. So the beta-lactamase, which is a penicillinase, it breaks the spawn. Once you have breaking of the beta-lactam ring, then this will lead to an inactive penicillin. Other examples as an application of what I said, we have, for example, beta-lactamases, okay, that are occasionally found in certain bacteria like Klebsiella pneumoniae and E. coli. And these are beta-lactamases, okay, they are modified in a way to become extended spectrum beta-lactamases, mean they act on a large spectrum of antibiotics, including the third generation cephalosporins like cefotaxime, ceftazidime, etc. So an example of the extended spectrum beta-lactamase that we call ESBL is the beta-lactamase CTXM15, which is encoded by the beta-lactamase CTXM15 gene. And this enzyme acts on these antibiotics that I mentioned, the third generation cephalosporins. So in our medical center, actually, we have a large proportion of E. coli and Klebsiella pneumoniae that are ESBL producing. Now, these organisms that are ESBL producing, okay, they are resistant to all third generation cephalosporins. So what you need to give them is another antibiotic called the carbapenems. And the carbapenems actually they have a spectrum activity on streptococci, Enterobacteriaceae, non-fermenters, anaerobes, so they have a broad spectrum of activity. And they consist of four main drugs like imipenem, meropenem, ertapenem, and doripenem that can be given. Now, ertapenem is the one that is the most susceptible to the effect of the beta-lactamase. That's why it is used also for screening purposes to know whether our bacterial population contains an ESBL. So these populations that are ESBL producers became actually resistant to ertapenem. Now the MIC is not 0.25, it's 2 microgram per ml. So to say that an organism is resistant to carbapenem, then the MIC, the minimal inhibitory concentration, should be 2 microgram per ml and above. And we have actually a good proportion now of carbapenem resistance. This is a little bit outdated. Now we have close to 4% of ESBL producing E. coli and about 10% of the ESBL producing K. pneumoniae that are resistant to carbapenem. Actually, are due to either the production of a carbapenemase, so it is a beta-lactamase that can hydrolyze carbapenems, and the most important ones that are currently available at hydrolyzing carbapenems is the KPC, Klebsiella pneumoniae carbapenemase, which is widely found in the US. And we have the OXA48, which is highly prevalent in the Mediterranean basin. And the M1 found in, originating from India, but this is not sure, since its name is New Delhi New Delhi beta-lactamase, metallo-beta-lactamase. So this one is actually the NDM. You have 1, 2, 3, and more currently that are found almost worldwide. Now also, carbapenem resistance may occur whenever a cephalosporinase, like a third generation cephalosporin, for example, ESBL, okay, with reduced outer membrane permeability or loss, or efflux pump, these can also cause resistance to carbapenems. So carbapenem resistance can be due either to a carbapenemase alone, or it can be due to an ESBL, okay, we call it cephalosporinase, an ESBL, but together with reduced outer membrane permeability and or efflux pump activity. Okay, so all these together can lead to carbapenem resistance. You have other types of mechanisms where microorganisms change their permeability to the drug, tetracycline for example. Microorganisms develop altered structural targets in the situation of erythromycin. Resistant organisms have an altered receptor of the ribosome. When the microorganisms develop altered metabolic pathways, and this is called in the situation of sulfonamide resistant bacteria. And 5, when we have microorganisms that develop an altered enzyme that can still perform its metabolic function, but it's less affected by the drug. And this is the situation in trimethoprim resistant bacteria, whereby the dihydrofolic reductase, okay, is inhibited less efficiently than trimethoprim susceptible bacteria. Okay, so in this situation, you have an altered enzyme that can still perform its metabolic activity. Now, multiple drug resistance and cross resistance. Pathogen can acquire resistance to more than one drug at a time. Common when you have plasmids, resistant plasmids that are exchanged. So on the plasmids that encode for different resistance genes, this will lead to resistance to multiple types of antibiotics. And this actually develops in hospitals, nursing homes, where constant use of drugs eliminates susceptible cells, and what remains are the resistant ones.